Welcome all to the next session, that is the third week of development. This is the week of gastrulation. Again, frequently asked in the MRCOG examination. We begin with our quote, Master MRCOG with concept-oriented clarity. Okay. Now, what happens in the third week? Okay. So, third week is one of the most critical period of the embryogenesis. It is characterized by the gastrulation, formation of the trilaminar germ disc, and establishment of the body axis. Okay, so once you hear the term gastrulation, trilaminar germ disc, think that we are in the third week of development. Okay, so second week we have seen it is a week of two. You have two cells, that is the epiblast and the hypoblast. You have seen the bilaminar germ disc. You have seen the two cavities. Now let us see the three-layer germ disc. Okay, so gastrulation, formation of the trilaminar germ disc, and the establishment of the body axis. This is what is going to happen in the third week. So, first event is a gastrulation. It begins by around day 15. Converts the bilaminar disc into the trilaminar germ disc. First, okay, so it will establish the ectoderm, mesoderm, and the endoderm. Cells migrate through a structure called as primitive stick. Okay, so a primitive stick develops on in this epiblast. Okay, so these cells, the migration is always through the primitive streak. Okay, this formation of primitive streak is very, very important, and the all cells are migrating through these primitive streaks. Okay, so coming to the formation of the primitive streak. Okay, so here is a primitive streak which is getting formed. It appears on the dorsal surface of the epiblast at the caudal end. Okay, so at the caudal end in the epiblast, this primitive streak develops. Actually, the caudal end itself gets developed by the formation of the primitive streak. Okay, there is no cranial or caudal till the development of the primitive streak. Okay, and this is just the disc, but the place where the primitive streak will form, we will call this as a caudal end. Okay, so it appears on the dorsal surface of the epiblast at the caudal end. So this is the epiblast, this is the hypoblast. Okay, this is the amniotic cavity, and this is the yolk sac cavity. Okay, so this is what we have seen up till now. Okay, now it consists of the primitive streak. Then there is a primitive node called as a Hansen's node, and there is a primitive pit. So you have a streak, then there is a node that is called as primitive node, and inside the node you have got a primitive pit, and through this primitive streak there is a migration of the cells. Okay, now with the formation of this primitive streak, there is a establishment of the craniocaudal axis. So this becomes the caudal part, and the part which is front of this becomes the cranial part. Then there is a right and left symmetry. Okay, and there is a dorsal and ventral axis. Up till now, this was there, but there was no symmetry as such. There was no dorsal ventral surfaces. There was no craniocaudal axis. Now, with this formation of the streak, you are getting craniocaudal axis, you are getting dorsal ventral surfaces, you are getting right left symmetry. So one of the important change is this formation of this primitive streak. Okay, so that is what is the importance of the primitive streak. Now let us see this in this figure. Okay, now this same thing what we have seen here. Okay, now how it will look. So this is the endometrium. Now the implantation is complete. Okay, now let us see, this is from the part of the uterine stroma. Okay, this is inside the uterine stroma. We are looking through the endometrium. Okay, so these are the two layers. Okay, epiblast is this, and then this is the hypoblast. Okay, so here it is the epiblast, here it is the hypoblast, here it is the amniotic cavity, and this is the yolk sac. Okay, so this is the yolk sac, this is the amniotic cavity. Okay, now what are the changes which are happening? So this is the place where this primitive streak is getting formed. Okay, at the caudal end of this. So at the epiblast, you are getting this particular primitive streak, and this is the hypoblast. Okay, so these cells will migrate through this primitive streak and will form an another layer. Okay, so here you see that this is the primitive streak, and there was a primitive node. See, this is the primitive node, and there is a primitive pit here, and through this there is a migration of the cells, and this is the hypoblast. So the cells will migrate from this place and will come in between. Okay, so cells differentiate at the primitive streak and migrate to form a middle mesodermal layer, as well as displace most of the hypoblast cells to form endodermal layer. The ectoderm forms from the epiblast. Okay, so now this bilaminar germ disk will get converted into this trilaminar germ disk. So this is the epiblast, then this is the primitive streak, and there is a proliferation of this cells, and this is getting formed into the mesoderm. Okay, this new layer will get formed here is the mesoderm. Then this hypoblast will get differentiated and will form the endoderm, and the epiblast at the end will get converted into the ectoderm. Okay, so these are the changes which are happening, and the bilaminar germ disk will get converted into the trilaminar germ disk. Okay, now coming to the formation of these germ layers. So endoderm, first cells to migrate through the primitive streak and replace hypoblast, forms the epithelial lining of the gut, respiratory tract, and the urinary bladder. Now the derivatives of each also they are asking very frequently in the examination. Derivatives of endoderm, derivatives of mesoderm, derivatives of ectoderm, that you should know. Okay, so the first cells to migrate through the primitive streak and they are replacing the hypoblast. So this hypoblast will get replaced by the cells which are migrating through this primitive streak, and the first cell to form is the this first layer to get formed is the endoderm, and that will replace the hypoblast and it will form the epithelial lining of the GIT, respiratory tract, and the urinary bladder. Then next is a mesoderm, that is the second wave of migration begins. It lies between the ectoderm and the endoderm. It differentiate into the this. Now you have got a new layer here, that is the mesoderm. Now this is this mesoderm will also have three parts. Okay, the part in the central and either side is a paraxial mesoderm that will lead to the formation of the somite. Then you have this intermediate part. You have this intermediate part that is called, that will develop into the urogenital system, and third is a lateral plate mesoderm that will form the body cavities. Okay, so this mesoderm will also have three parts: the paraxial part, the intermediate part, and the lateral part. Okay, lateral part will also get split into two: the somatopleuric and the splanchnopleuric. Okay, so this somatopleuric and splanchnopleuric is different than what we have seen extraembryonic. They are different, so do not get confused. Now we are talking about the intraembryonic mesoderm. In the initially we have seen the extraembryonic mesoderm. That was a different. Okay, now this is the intraembryonic mesoderm. It will have three parts: the paraxial part, the intermediate part, and the lateral part. Okay, so this paraxial mesoderm will lead to the formation of the somates. The intermediate mesoderm will lead to the development of the urogenital system. Again, here is an SBA. Please remember this. And lateral plate mesoderm will lead to the formation of the body cavities. Okay. Now the last layer is the ectoderm. So remaining epiblast will form this ectoderm, and it will form the central and peripheral nervous system, epidermis, and its derivatives. Okay. So these are the structures which are getting formed. Okay. Let us see the same thing here. Okay, so this is the developing embryo. This is the yolk sac, which is lined with the endoderm. This is the yolk sac. Okay, now this is the amniotic cavity. Okay, so this is the amniotic cavity. This is the amnion. Okay, this outside is the endometrium. Now three layers are formed: the ectoderm, endoderm layer, mesoderm layer, and the ectoderm layer. Now let us see the derivatives. Endoderm layer becomes the digestive system, liver, pancreas, and the lungs, the inner lining. Okay, inner layers of the lung will be formed from the endoderm. Now mesoderm will give rise to circulatory system, lungs, the epithelial layers of the lungs, then skeletal system, muscular system. So musculoskeletal system, the circulatory system, and epithelial layer of the lung will be formed from the mesoderm. Okay, while ectoderm will give rise to hairs, nails, skin, and the nervous system. Okay, so these are the derivatives of the mesoderm, ectoderm, and the endoderm. Okay, same is explained here. You have the endoderm or the internal layer. The lung cells or the alveolar cells are getting formed. Thyroid cells are coming from the endoderm. Digestive cells, the pancreatic cells are coming from the endoderm. Now mesoderm, musculoskeletal system, you remember. Okay, you have got the cardiac muscles, skeletal muscles. Tubules of the kidney are coming from the mesoderm. RBCs are coming from the mesoderm. Okay, musculoskeletal and the vascular. Okay, smooth muscles, cells of the gut are also coming from the. So all muscles you are getting, the RBCs you are getting, and the tubules of the kidney you are getting from the mesoderm. From the ectoderm, nervous tissues and the skin. Okay, skin cells of the epidermis, neurons, and the pigment cells, they are coming from the ectoderm layer. Okay. Now next step is a formation of the notochord. Okay, next step is a formation of the notochord. Okay, so here is, you see in this picture is a notochord which is getting formed. Okay, and here is a mesoderm. Okay, and this is the ectoderm. Okay, this is the ectoderm, this is the mesoderm, and here in the mesoderm you see a notochord. Okay, so this notochord is derived from the mesoderm. It develops from the cells migrating cranially from the primitive node. Okay, so there is a migration of the cells from the primitive node which is getting which is forming in the new layer, that is the mesoderm. So this notochord develops from this mesoderm from the cells which are migrating cranially from the primitive node. Okay, so here at the here there will be primitive node, and cranial to the primitive node these cells will migrate and will form the notochord. Okay, so what does it do? It defines the primitive axis, then it induces the neural plate formation. It persists as a nucleus pulposus of the intervertebral disc. So this is also a frequently asked question. The remnant of notochord is the persistence in the adults as a nucleus pulposus of the intervertebral disc. So we have seen that there is an ectoderm, there is a mesoderm which is getting formed. In the mesoderm, this notochord gets formed. Okay, cranial to the primitive node. Okay, the cells which migrate cranial to the primitive node will form this notochord, and with this formation of the notochord, this ectoderm will have the induction further for a formation of this neural plate. You see that neural plate is getting formed into the ectoderm. So this mesoderm and the ectoderm is overlying the mesoderm. Then there is a induction in this ectoderm for the formation of the neural plate, and these are the borders of the neural plate which are called as neural plate borders. They are very specific in nature. That is also you should remember what it will form. Okay, so this neural plate will further invaginate and it will form the neural plate, and this neural crest border will join to each other, and this neural crest cells, this border of the neural crest cells will form the components of the peripheral nervous system. This all development is going to be very important from the examination point of view. You'll have lots of SBS. Okay, so this ectoderm, the invagination, then this neural cross, there is a joining of these two will be forming the neural tube, and this neural crest, that is the neural plate border, will now labeled as a neural crest because of this invagination formation. This neural crest cells will form the components of the peripheral nervous system. Okay, now you have got a neural tube. Now you have got these peripheral nervous system components, and you have got this notochord, and this will be the your epidermis or the ectoderm. Okay, now let us see. So neuroectodermal tissue differentiate from the ectoderm and thicken into the neural plate. Okay, so the notochord is formed. With this notochord formation, there is a neuroectodermal tissue differentiate from the ectoderm and thicken into the neural plate. The neural plate border separates the ectoderm from the neural plate. So these are the neural plate border. This will separate this neural plate from the rest of the ectoderm. Okay, the neural plate bends dorsally with the two ends eventually joining at the neural plate borders, which are now referred to as neural crest. Okay, so this neural plate, okay, bends further, it bends, and the two ends eventually join each other. Okay, now what happens further? The closure of the neural tube disconnects the neural crest from the epidermis. Neural crest cells differentiate to form most of the peripheral nervous system. So these neural crest cells will form the peripheral nervous system. Okay, the notochord degenerates and persist as the nucleus pulposus of the intervertebral disc. Other mesoderm cells differentiate into the somites and the precursors of the axial skeleton and the skeletal muscles. So the notochord is getting formed. This notochord will induce this formation of the neural plate. This neural plate will then form the neural tube, and this is the formation of the components of the peripheral nervous system. Later on, this notochord will disintegrate. However, in the adults, it will remain as a nucleus pulposus of the intervertebral disc. Okay. Then the neurulation begins by around late third week. Neural plate forms from the ectoderm. There is a thickening of the notochord. Precursors of the This is the precursor of the brain and the spinal cord. So this neural tube will further lead to the formation of the brain and the spinal cord. Okay. Now there's a formation of the somates. So we have seen in the mesoderm, the first is getting formed is a notochord. Now further what is getting formed in the mesoderm is the somate. Okay, so these are the somates which are forming. Okay, so this is the notochord, this is the rest of the mesoderm, and from there you are getting these somates. Okay, these are the somates which are getting formed. So these somates, they are derived from the paraaxial mesoderm. They appear in the craniocaudal sequence. Each somite differentiate into sclerotome, myotome, and dermatome. Sclerotome will give rise to vertebrae and ribs. Myotome will give rise to skeletal muscles, and the dermatome will give rise to the dermis of the skin. Okay, so these are the somates which are getting formed into the mesoderm. They are further differentiating into three: sclerotome, myotome, and the dermatome. Sclerotome will give rise to vertebrae and ribs. Myotome will give rise to skeletal muscle, and dermatome will give rise to the dermis of the skin. Okay. Now, next is the development of the intraembryonic coelom. Okay. Up till now, what we have seen is the extraembryonic mesoderm and the extraembryonic coelom. That is different. Now we are talking about the in the embryo. Okay. So in the embryo, there is a development of the intraembryonic coelom. This lateral plate mesoderm splits into two: somatic or the parietal mesoderm and the splanchnic or the visceral peritoneum. So this mesoderm, as I mentioned, this is the middle layer. That is the mesoderm. Okay, so centrally there is a notochord formation, then there is a paraxial mesoderm, then there is an intermediate mesoderm, and then there is a lateral plate mesoderm. This lateral plate mesoderm also splits into two. Okay, one is a somatic or the parietal mesoderm, and another is a splanchnic mesoderm. Okay, so this lateral plate mesoderm will also get split into two. And what does it form? It forms the pericardial, pleural, and the peritoneal cavities. Then formation of the blood vessels and the heart primordium. Then vasculogenesis begins. Blood islands form in the yolk sac, intraembryonic mesoderm, and the heart tube formation begins by the end of the third week. Okay. Then the allantois formation. It appears as an outpouching from the yolk sac. It contributes to the umbilical vessels and the urinary bladder. Okay, that is the role of the allantois. Okay. So from the examination point of view, most sensitive period of teratogenesis is this third week of development. Failure of the primitive streak regression will lead to the sacrococcygeal teratoma. This also you should remember from the examination point of view. Failure of the primitive streak regression, okay, will lead to the sacrococcygeal teratoma. Failure of gastrulation will lead to caudal dysgenesis, and notochord remnants will lead to the chordoma. Okay. So most of the notochord will, what will happen? It will disintegrate. But later on in adults, it is remaining as a only the nucleus purposes of the intervertebral disc. But if it persists, the notochord, then it will lead to the formation of the chordom. Okay. So if you see in the flowchart format, this third week of development, the primitive streak is getting formed. So first is the formation of the primitive streak by around day 15. The epiblast cells proliferate and form a linear groove on the dorsal surface that marks the craniocaudal axis of the embryo. After that, there is a gastrulation. That is the formation of the three layers now. Epiblast cells will migrate through this primitive streak, creates a three germ layers. The endoderm, first migrating cells, displace the hypoblast, then mesoderm, that is the middle layer, and finally it is the ectoderm, which has the cells lining the epiblast. Okay. Then by this time, the trilaminar just disc is getting formed. So the embryo transitions from the bilaminar to the trilaminar germ disc. Okay. Then there is a notochord formation. Cells from the primitive node migrate cranially, and the notochord indicates the neural plate formation. The early neurulation begins. Neural plate appears within the ectoderm above the notochord. Then there is a somite formation in the mesoderm by around day 20. The paraxial mesoderm segments will lead to the formation of the somites. Then there is an intraembryonic coelom and the hard primordium. The coelomic spaces form body cavities. The vasculogenesis will start and the primitive heart tube will begin formation. Okay. So this Paraxial mesoderm will lead to the formation of the somites that will lead to the formation of the vertebrae, skeletal muscles, and the dermis. The intermediate mesoderm will give rise to kidneys, gonads, genital duct, and the adrenal cortex. And the lateral plate mesoderm will give rise to body cavities, heart, and blood vessels. Okay, so this is the fate of the mesoderm. You have got a paraxial mesoderm, you have got the intermediate mesoderm, and the lateral plate mesoderm. This lateral plate mesoderm will form the body cavities, heart, and the blood vessel. That finishes the third week of development. Okay, thank you. Uh, just a moment. Can you see the slide now? Can you see the slide? You are able to see
Yes,
the
sir.
slide?
Yes,
Yeah.
sir.
Yeah, this slide was not visible properly. So you have this notochord. Okay, you have this neural tube. You have this notochord. Okay, and this is the middle layer, that is the mesoderm. This mesoderm gets divided into three: paraaxial, intermediate, and the lateral plate. So this is the paraaxial, this is the intermediate, and this is the lateral plate mesoderm, which is further getting split into two. Okay, now let us see the derivative of it. This is the intermediate mesoderm, which will give rise to kidneys and gonads. Now this is the notochord, cord of mesoderm. Okay, that will lead to the formation of the notochord, and that will remain in the adult as a nucleus pulposus of the intervertebral disc. Then you have got a paraaxial mesoderm. Okay, so this paraaxial mesoderm will lead to the formation of the head, the somites. The somites are further sclerotum and the dermatomyotome. Okay, the sclerotum will lead to the formation of the vertebrae and ribs. Then endoderm, that is the endothelial cells and the dorsal aorta. Arthroderm, that is the vertebral joints, and syndoderm, that is the tendons. Okay, this dermamyotome will form the myotome, that is the skeletal muscle, and the dermatome, that is the dermis and the skeletal muscles. The lateral plate mesoderm will be splanchnic mesoderm, somatic mesoderm, and the extraembryonic mesoderm. Okay, so these are the different parts which are getting formed from this mesoderm and the notochord, etc. Thank you.